All right, let's talk about the blood. Now, there are several different types of blood cells. The first one we think about is the red blood cell, also called erythrocyte. The function of the erythrocyte is to carry oxygen to the cells of the body. It also helps carry the carbon dioxide back to the lungs, but let's think about the erythrocyte as carrying oxygen to the cells of the body, the central nervous system. Here you see it in it's round, of course, and it's got this dimple in it on both sides, and that increases the surface area. This cell is basically just a sack of hemoglobin. There's no nucleus, there's no organelles. It's just a plasma membrane filled with hemoglobin, which is the protein that carries oxygen. The hemoglobin also contains iron, and that is part of its molecular structure. So that means that they can't reproduce and they need to be replaced from the bone marrow. So the shape is on both sides, and this is what makes the blood red. Some places will list that the function of the red blood cell is to make the blood red. We don't care what color the blood is. We care that it carries oxygen. So the function is to carry oxygen. And we also see some white blood cells and maybe some little bits of platelets. So if we take the blood from someone, and you've probably had that done, and run it in a centrifuge, we separate out the blood into its components. And remember, blood is a connective tissue. It's a fluid connective tissue. The living cells are the red blood cells. They are suspended in the fluid matrix. So the plasma is the non-cellular matrix. And the red blood cells are going to centrifuge to the bottom because, of course, they contain that hemoglobin, that heavy hemoglobin with the iron. And so they're going to come to the bottom. Right above that, we have what we call the buffy coat, and that's where the white blood cells are. When we use the small capillaries, we can just barely see that. Well, really, we can't see it with the tiny capillaries, but so it's a very thin layer. And then above that, we have the blood plasma, and this is the water, the proteins, the nutrients, the hormones, waste products. This is the plasma. So these are also important things to be carried in the blood. For example, nutrients that we've gotten from the digestive system and we're carrying to the cells to be used. So here we see a normal amount of packed red cells. So this hematocrit is also called the packed red cells. And so for women, we have about 37 to 47% normal. Percent being measured, this distance over the whole divided by the whole difference. This is a fraction of the entire length. Men have a little bit higher hematocrit, 42 to 52. If it's low, that is anemia, and a person is not going to be able to carry the oxygen that they need to the cells. They're going to be tired. They're going to have a hard time thinking. Their muscles aren't going to get, the muscles and brain are not going to get enough oxygen. Now, if we have too many red blood cells, this could be a sign of dehydration. This blood is going to be so thick that it's going to be hard for the blood to pump, for the heart to pump that blood through the blood vessels. Sometimes when we take blood samples in the summer, some of the guys will have, you can hardly get that blood to come out. And they need to drink some water, right? They're getting a little dehydrated. So you don't want it to be too thick. So this is a summary of the components of the blood. So remember, we've got the plasma or fluid portion, and that has predominantly water, plasma proteins. We've got the hormones, waste products, and so forth, nutrients. And then we've got what we call the formed elements. And the reason we don't just say cells instead of formed elements is that the platelets are not actually cells. The platelets are fragments of a larger cell. So in these formed elements, we've got the red blood cells, the white blood cells, and the platelets. And the function of the platelets is going to be in blood clotting when we're cut. The platelets are going to play a role in blood clotting. We'll talk about blood clotting a little bit. You're going to get a whole lot more about that in physiology. And we'll cover the white blood cells again in a little bit. Most of those white blood cells are fighting pathogens and defending the body. All right. So this is a different source. This is OpenStax. This is not from your Pearson book. I don't need you to know all of this. I do want you to know that the blood formation occurs in the bone marrow. It occurs in the red bone marrow, right? So that's in the spongy bone. And I want you to be able to recognize the cells with what we get at the end, right? So there's our platelets. So follow along in the book. Look at these. You want to be able to recognize a white blood cell either in an image or on a model, and you need to be able to tell me what it is and what its function is in a general way. You don't have to go into a whole lot of detail, but you need to know its function. All right, we'll keep looking at those, but you don't need to know at this point all of these steps in between. All right, and here's another look at some of these cells, right? So we recognize them by the lobes of the nucleus, by their size, by the granules in the cytoplasm, and those granules are just related to how the cell picks up stain. So we've got lobes of the nuclei, the granules, the overall staining, and the size is how we recognize these and sort them out. So here I want you to be able to recognize them. I want you to know their function. There's some other detail here, but I want you to know to recognize them by image, know their function, know the name of them, and their prevalence, which ones are the most common. All right, here's our red blood cells. We talked about them a little bit before. They're biconcave discs. So this is, it's kind of like a donut with a little membrane in the middle. All right, so that gives us our surface volume to facilitate gas exchange. It also makes them a little bit flexible, kind of like a soft frisbee in a way. So they can go through the blood vessels easily, right? They can bend and flex through the capillaries. So a capillary is about the diameter of one red blood cell. All right. All right, here's our hemoglobin molecule. I want you to know that it contains iron, here shown as Fe, right? And it's made of 4 subchains, 4 protein subchains, each of which contains that iron molecule. You'll learn more about this in physiology. So that's one molecule, right? That's one molecule of hemoglobin right there. And that red blood cell is just filled with this protein. Right there you see that iron molecule in the middle. Oh, there's some nitrogens around it. Okay. Erythrocyte life cycle. So this one I do want you to know a little bit about the life cycle. So it's formed in the bone marrow. It loses its organelles, so it is not able to reproduce. Once that red blood cell is out in the bloodstream, cannot reproduce. It doesn't have mitochondria, so it cannot use the oxygen that it's carrying, right? It can't eat the profits, right? It's just carrying that oxygen. And they last about 120 days, and then they're recycled in the spleen where the body tries to get that hemoglobin, that iron molecule back, right? So some of this is processed as bilirubin, some comes back and is reused in the bone marrow. In the case of sickle cell anemia is a genetic disease with a single point mutation in that hemoglobin molecule, and that single mutation causes the red blood cell to have this completely different shape. And notice that this is not a nice shape for going through capillaries, right? The round cell is going to slip right through a round cell. This is pointed. This cell can jam up in the capillaries and can be quite painful. All right. So this is an illustration of the white blood cells fighting disease. You will look at this more in physiology. So I'm not too worried about this right now. All right, here is a better look at some of those white blood cells. Now, this staining, the color that we see here is a chemical stain. It's not the color that they are in our bodies. This is how they take up stains so that we can see them in the light microscope. So there we see that neutrophil, and we see several lobes in that nucleus, and it does have some small granules and a little bit of kind of staining lilac. There's our eosinophil. We've got two lobes there in the nucleus. There, the basophil looks pretty blue, stains dark blue. It's telling us two lobe there. It kind of looks one lobed. Right, these are fairly easy to distinguish. All right, here are the platelets. So they are parts broken off of a cell called a megakaryocyte. So megakaryocyte's great big cell with things flaking off of it. So those platelets are little pieces that are going to form the blood clot. Right, so we'll talk a little bit more about that in a second. All right, here's our basophil, eosinophil, neutrophil, monocyte, and lymphocyte. So make sure that you see those in lab and make sure that you can identify them. All right. So this is our summary of clotting. This you will also get much more detail in physiology, so I'm not going to spend a lot of time on it or expect you to know it. I'll say that it is a multi-step process, and it uses several different enzymes, right? And you get this blood clot with fibers, platelets, with red blood cells, and proteins in it. So I'm not too worried about that right now. All right. This you will probably get in physiology as well, but I'm going to talk about it because you can kill a person, and this is really bad. So in blood typing, we have different blood types, and they refer to a protein molecule that's on the surface of the red blood cell. You can have protein A, protein B, you can have both, or you can have neither. There's another protein called the RH, also called D. In this place, it's this card or this slide, we're seeing it as D. So let's just talk about A and B. In this case, we've put a sample of blood. Let's switch to a different color. So we put a drop of blood on each one of these places, just a drop of blood from one person, and we've added antisera and stirred it around and let it sit for a minute or so. And we can see here, this is called agglutination. The thing we see here is agglutination. And that indicates to us that this blood reacted with the antisera A. So it had protein A, right? So we can say this person is type A. We look at this place with antisera B, and it's just smooth. It's dried out a little bit, but it's smooth. So they are not B. We can look here and see that again it agglutinated, and so they had this protein D, also called RH, also referred to as just positive or negative. This, I agree, is very confusing. So this would be called type A positive. So that would mean that this person had protein A and they had the Rh protein. Make sure that you understand this before you blood type people. Here is a graph showing these blood types. So here is a person with just that A, just B, A and B, neither, and that's O. And that's inherited separately from the Rh factor. So this O could be positive or negative. The AB could be positive or negative. B can be positive or negative. A can be positive or negative. So different sources are going to be describing that differently. They could describe it as D, as RH, or as positive and negative. You need to be familiar with all of those. All right, so this was from OpenStax. This was very short, and I encourage you to read the OpenStax or the Mastering digital text on the blood.